Great. Thanks everyone for joining us today. So today we have Sanjay Curtis, who's a postdoctoral researcher at the Liverpool School of Tropical Medicine, and he studies genomic surveillance and the molecular mechanisms of insecticide resistance in Anopheles gambiae. So today, Sanjay is going to talk us through his paper about RNA-Seq-Pop, which is a workflow which provides end-to-end analysis of RNA sequencing data set. So this workflow does a lot of stuff, including performing quality control, differential expression analysis, and calling genomic variants. But also, it has a lot of other options, which includes things that we've been talking about in previous workshops. For example, summaries of genetic variation and population structure and genome-wide selection scans, which you are quite familiar with now. So thanks again, Sanjay, for joining us, and I'll leave the floor off to you.
Thanks, Anastasia. Hi, everyone. Welcome to the fifth journal club of the PAMCA MalariaGEN workshops. As Anastasia said, I'm Sanjay. I'm a postdoc at the Liverpool School of Tropical Medicine. And today I just wanted to talk about this paper that we published last year, RNA-Seq-Pop, exploiting the sequence in RNA sequencing. A Snakemake workflow reveals patterns of insecticide resistance in the malaria vector Anopheles gambiae. And so this journal club will be a little bit different to the previous journal clubs, and also a little bit different to the PAMCA workshops so far, because we're focusing today on the analysis of transcriptomic data. So specifically RNA sequencing data rather than whole genome sequencing data. Just as a heads up, I don't think this talk will take the full hour. So as we know, as we've seen in the workshop so far, insecticide resistance is an escalating problem in malaria mosquitoes that's sort of threatening to curb the gains that we've made in malaria control in the past 15 or 20 years. And so the mechanisms of insecticide resistance can generally be split out into 4 different categories. These are target site mutations, which we saw in workshop 1, such as KDR or ACE1. Metabolic resistance, which we saw in workshop 2, which can be caused by a number of different enzymes, such as cytochrome P450s, esterases, GSTs, and so on. Cuticular resistance, which involves the thickening or hardening of the cuticle, and also behavioral resistance as well, whereby mosquitoes might avoid contact with insecticides. And so, although in recent years, as you guys have already seen, whole genome sequencing has sort of appeared as a really useful tool to get to the bottom of these mechanisms of resistance. Prior to whole genome sequencing, so talking over the past 20 years, we primarily discovered these genes, particularly metabolic and cuticular genes, through transcriptomic studies, where we were comparing gene expression between a susceptible and a resistant strain of mosquito to identify genes that might be involved in that phenotype. So just very quickly, what is transcriptomics? So when we talk about transcriptomics, we're talking about measuring gene expression across the whole transcriptome. So for all genes in the Anopheles gambiae genome, we'd like to measure expression and compare expression between 2 conditions. And so why do we want to do this? Well, we know that the overexpression of certain genes can lead to insecticide resistance. So particularly with metabolic resistance, overexpressing an enzyme such as cytochrome P450 might mean that a mosquito can metabolize and remove more of the insecticide, leading to its survival. And so we saw in workshop 2 as well that CNVs, whereby a gene is duplicated or triplicated or so on, that's one mechanism within which a mosquito can increase expression of a gene. And so in the early days, around 2005, probably for the next 5 or 10 years, microarrays were used as the main technology for this. And nowadays they've kind of been replaced now, particularly by Illumina RNA sequencing. And so in this in this paper, we present the computational workflow called RNA-Seq-Pop, which is written in Snakemake. And as Anastasia mentioned, it's a computational pipeline to analyze Illumina RNA sequencing data of any organism. So although today I'm going to be focusing on analysis of Anopheles gambiae and focusing on insecticide resistance, you can apply RNA-Seq-Pop to any organism and to any phenotype. And so, as well as performing standard transcriptomic analyses, such as differential expression, RNA-Seq-Pop also calls and can analyze genetic polymorphisms and extract some population genomic signals, which we're going to look into today. If you'd like to visit the GitHub, the link is here, and the documentation as well. So I mentioned it's written in Snakemake. In case you're unfamiliar with what Snakemake is, Snakemake is a workflow management system. So if you may have heard of Nextflow, this is a very similar tool. And the idea is that these tools help researchers to create reproducible and scalable pipelines. And so they have many sort of useful features within them, but they allow you to scale your pipelines to servers, it could be a personal computer, clusters, or the cloud without modifying the underlying code. So all of that is handled by Snakemake or Nextflow itself. And importantly, all the software and dependencies for the pipeline can be installed automatically using Conda, which is obviously quite important, that users don't need to install every bit of software themselves. So in this paper, we were focusing on a strain that we had collected from Busia in Uganda. And so this was going on at the time of a really large-scale bednet trial, PBO bednet trial in Uganda called the lineup trial. And so the idea was that we had a strain from Busia, which was collected by my colleague Ambrose Aruni. And so we wanted to look at mechanisms of resistance in this strain. And so we thought we would do an RNA sequencing study. And so we had a G24 Busia strain, which had actually lost a lot of resistance when we were looking at it. So actually, at the point where we started the study, it was 100% susceptible to deltamethrin, and 97% showed 97% mortality to permethrin. So it had lost most of its resistance while it had been in the lab and it hadn't been selected. So we then, over 4 generations, each generation we selected with WHO tubes to deltamethrin, and that resulted in a third strain, the G28 Busia selected strain, which was moderately resistant to deltamethrin, showing 70% mortality, but showed even higher resistance to permethrin with 30% mortality, which is quite interesting in itself because we were selecting with deltamethrin, but the permethrin phenotype increased more than the deltamethrin phenotype. And so this map on the right just shows the locations of Busia and also Kisumu. So we used the Kisumu lab strain as a sort of third group. And so I think many of you will be familiar with this strain. It's been in the lab for a long time. I think it was originally established in around 1975. And so Kisumu is susceptible to all insecticides. So we performed sequencing with NovaGene, sequencing around 840 million reads in total. These libraries were sequenced to quite a high depth. So I think for each biological replicate had around 45 million reads that was successfully mapped to the pest reference genome. And we also used 75 paired end reads, which seems pretty much standard for RNA sequencing at the minute. So RNA-Seq-Pop uses a number of tools to assess quality, including FastQC for the read quality. And then we'll use a tool called MultiQC, which wraps all of the QC info from various tools into a HTML report to make it quite easy to navigate and for the user to view. One thing prior to differential expression that the pipeline will do is to, perform principal components analysis on the normalized read count data. And it will also perform heat maps, sorry, not heat maps, hierarchical clustering as well. And the idea behind this is we want to see that our biological replicates cluster together. So we can see, if we look at this PCA here on the left, we can see that each condition does indeed cluster together. And this gives us a bit more confidence in the underlying data. And this is also mirrored in the hierarchical clustering as well. So that's positive. RNA-Seq-Pop will perform differential expression analysis, both at the gene level, using Callisto and DE-Seq2, and also at the isoform level as well, using a tool called Sleuth. It will produce the traditional kind of plots you see from transcriptomic analyses, such as volcano plots, which we have an example of on the right-hand side here. And also you can provide RNA-Seq-Pop with a list of genes that you're interested in, and it will produce heat maps of those genes as well. I'm not going to go into too much detail about the specific results from the differential expression analysis from this paper, as I'd rather focus on the sort of SNP calling aspect, which is slightly novel in RNA-Seq-Pop. But there was a number of detoxification genes that were overexpressed. There were a lot of P450s that were sort of mildly overexpressed. The chemosensory protein SAP2 showed a really high overexpression when we compared the selected Brucia strain to the Brucia parental strain. And also there was a cluster of carboxylesterases which were all overexpressed in these comparisons as well. And so another thing that the pipeline will do is summarize the results of the differential expression analysis for different gene families. So for example, we can summarize it by cytochrome P450s or GSTs, odorant binding proteins, and so on. And this is based on PFAM domains. So we extract a list of genes based on their PFAM domains. And so this plot on the right just shows an example of that for GSTs. So we can see we have sort of each gene on the y-axis, and the blue sort of heat maps are representing the normalized read count data. So where there is a very faint, that means a gene is very lowly expressed, and where it's very dark, that means these genes are very highly expressed. And similarly, on this left-hand side, this shows each differential expression comparison that we did. And gray means that there was no differential expression in that gene in this comparison. Green means that gene was overexpressed, and purple means that gene was underexpressed. And so, yeah, the pipeline does quite a few additional smaller differential expression analyses, which I'm not going to touch on in any detail, but things like gene set enrichment analysis for different Go terms or KEGG pathways. It can produce Venn diagrams between differential expression comparisons. So if you have two different differential expression comparisons and you want to see are the same genes being upregulated within these comparisons, we can produce Venn diagrams for that. We can also see if we have multiple differential expression comparisons, if we want to see if genes show the same pattern across both of those comparisons, we can look into that. And finally, we can also see if our differentially expressed genes are in the locality of a selective sweep in the AG1000G phase 1 data. So this last one is a little bit out of date because it's using phase 1 data, but we can update that. So yeah, what I really wanted to focus on with this talk is on SNP calling with RNA sequencing, because this is really what makes RNA-Seq-Pop a little bit different to other pipelines that might analyze transcriptomic data. So this is something that's quite rarely performed in RNA sequencing data, and there may be good reasons for that, but in our field, we have a situation where many groups, many people are doing these kind of transcriptomic studies, particularly focusing on insecticide resistance, trying to find genes that are involved in a phenotype. And so we have lots of these RNA sequencing data sets, and most of these studies are being done just thinking about differential expression only. So we have lots of these data sets, and often we also have no matching whole genome sequencing data from the same populations or cohorts. And by actually using the sequence data within our RNA-seq data, it can really help us bridge the gap between our transcriptomic studies and the whole genome sequencing studies that we're doing, the kind of studies that you guys, and the kind of data that you guys have already been analyzing in the first 4 workshops. As I said, we're already doing this for differential expression purposes. So all of this extra information that we're getting by calling SNPs, it's free. You know, we're really making the most out of our data, which is quite nice. And the other thing is that transcriptomic data is generally quite noisy in itself, and so we often have, you know, thousands of differentially expressed genes as well. And so, you know, identifying which of those 1,000 genes might be important is still really difficult, even after performing a transcriptomic study. And so if we can integrate some SNP data, that can provide additional evidence into whether a gene might be involved in resistance, that could also be really useful. So there are some caveats to calling SNPs with RNA sequencing data. And so first of these is that, well, first of all, we can only call SNPs in regions of the genome that are actually expressed, because this is RNA sequencing data. We're essentially trying to sequence messenger RNA only, and so only parts of the genome that are actually being transcribed into messenger RNA, we will only observe those. Now that in itself is maybe not too bad. I mean, this is the premise of exome sequencing. So in exome sequencing, it's a bit like whole genome sequencing, but we just focus on exonic regions, basically. And so you could say that, well, we are at least focusing on the most important, what might be the most important regions of the genome. Another caveat is that the allele frequency that we see in RNA sequencing reads may not actually reflect the allele frequency in DNA. And one major reason for that is because of a phenomenon called allele-specific expression. So allele-specific expression is a process that happens. For example, a mosquito has 2 alleles at any given locus, but for some reason, only one of those alleles is actually expressed. And so in the case of RNA sequencing, we would only observe the expressed allele. We would not know that there is not another allele present. There are other reasons that could lead to false positives, so things like RNA editing. So RNA editing is a process in which after transcription, RNA is sort of edited into a different base. We would not be able to observe that within the RNA sequencing data directly, and so it might be called as a mutation. And also, we have really high coverage genes in RNA sequencing data. You will have some genes which have hundreds of thousands of reads mapping to them, which is very different from whole genome sequencing data, where typically coverage is much more even across the genome. And so this in itself could also cause some errors potentially. So we do call SNPs with a program called Freebase, and we remove any SNPs that have low depth. So if there are only a few reads mapping to that SNP, we'll remove it. We will remove SNPs with low quality scores. And also, if a SNP is only found in a couple of biological replicates, we will also remove that as well. And so we perform this on the BUSIA data, and we find that, as you would expect, SNPs are enriched in transcribed regions of the genome. So this diagram here shows essentially the proportion. At the top, we have the proportion of the reference genome that is within each genomic feature. So, for example, we have 54% of the AGAM-P4 reference is intergenic. And what we find is when we look at SNPs in the data set, we find most of these SNPs, the vast majority, so 84%, are found in genic regions, which we would expect. We do still find a small amount of SNPs falling in intergenic regions, which could be due to misannotation of the reference genome. It could also just be due to expression of parts of the genome which are not actual genes. So this makes sense anyway, that we're finding this enrichment. Once we have that SNP data, we can perform another PCA on the genotype data itself. What we tend to find is that this mirrors the principal components analysis that we do on the normalized count data. We can also look into genetic diversity. So we can calculate nucleotide diversity, pi, and also Watterson's theta. And when we did this with the 3 strains in the BUSIA data, we found that the Kisumu lab colony had the lowest genetic diversity of the 3 groups. And that made complete sense because the Kisumu colony has been established since, I think, 1975. And so we know that it would have been through strong bottlenecks in a lab and should have produced diversity. Similarly, we find that the BUSIA parental strain has the highest diversity of the 3. And this makes sense because it had not been under any selections. And it had also spent, out of the 3 groups, the least time in colony. And the G28 strain was somewhere in the middle of that. So I mentioned, obviously, we are interested in insecticide resistance. And one thing that we can do then is calculate allele frequencies at mutations, which we know are important in insecticide resistance. So this figure on the right just shows allele frequencies at a sort of curated list of mutations, which we know are important, and their allele frequencies in each group. And so what we can see is we have the BUSIA parental strain. We can actually see that we knew that this strain had low resistance low or no resistance when we started the selections. And we can actually see that the VGSC mutation L995S, which we know is present in Uganda at quite a high frequency, was at around 25% frequency. And after 4 generations of delta methylene selections, this actually increased to 100%. So pretty much to fixation potentially. And so this makes complete sense. We know that this mutation is important for resistance to pyrethroids. So we would expect it to increase in frequency over the period of delta methylene selections. We also found an increase in a marker called CYP4J5, the L43F mutation, which has been associated with pyrethroid resistance in Uganda in the past. So that was also a kind of confirmatory result. And we also saw a big increase in a marker in GST2, L119V. So GST2 is generally more associated with DDT resistance, but it is also potentially could cause pyrethroid resistance as well. We see a big increase in that marker. The last interesting thing that we found with this is that we found a mutation I114T, which is well known to be important for DDT resistance. We found this mutation in Kisumu, which was surprising because Kisumu should be a completely susceptible colony to different insecticides, and so we did not expect to find any resistance mutations in Kisumu. The kind of interesting thing about this is that then we went back and looked at phenotypic data from Kisumu, which was stored by LITE, the Liverpool Insecticide Testing Establishment in Liverpool. And we actually found that every couple of years, you find a low level of DDT resistance in Kisumu sort of crops up. And so whether it could be that the reason for this slight cropping up of DDT resistance is because this mutation is in Kisumu is possible. Why that mutation may have ever got into Kisumu in the first place, we'll touch on later. So we can also look at genetic differentiation across the genome. So I think you guys have already done some work on genome-wide selection scans. or if not, you definitely will do soon. And so using the called SNPs, we can calculate genetic differentiation across the genome between each experimental treatment group. And so the idea is we want to identify regions of the genome which differ between phenotype, which might indicate there's something important in that region. And so this plot on the bottom just shows FST, so a measure of genetic differentiation between the G24 Busia parental strain and its G28 selected offspring. And if what we can see is like there are a few sort of really large peaks of FST, and we find that there's one really large one around the VGSC, and we also find in the region of where the 2L8 inversion is, we see a sort of elevated region as well. There are also potential minor FST peaks around SAP2, which we know was overexpressed, and also GST2. And so if I just go back to that plot of allele frequencies, this kind of makes complete sense, because we know that between the Busia parental strain and the Busia selective strain, we've seen a huge shift at the VGSC. So in terms of the haplotypes that are, in these two strains, there's been a big shift to one specific haplotype probably. And then we see really high genetic differentiation at that locus. Similarly, we saw a GST2, there's a sort of big shift, and whether or not you would call this a peak, I'm not sure, but there does seem to be potentially something around GST2 as well. And we'll touch on 2LA in a moment. So this is potentially interesting anyway. It's quite nice that we're seeing these shifts in allele frequencies, which we know that they are important for resistance. And we're also seeing, when we look across the genome, we're seeing these big signals at the same loci. So I mentioned the 2LA inversion. There seems to be a sort of extended region of high genetic differentiation. Something else that the pipeline can do is actually karyotype our samples for chromosomal inversions. So in case anyone isn't familiar, the Anopheles gambiae scintillato harbors a number of chromosomal inversions, which are regions of the genome which have essentially flipped, they've inverted, and these are present at varying frequencies depending on where you are in the range of Anopheles gambiae. And initially they were, for example, the 2LA inversion, which is found here on chromosome 2L. It was found to be important for desiccation resistance and aridity tolerance, and more recently has been found to potentially be involved in plasmodium infection and also in insecticide resistance as well. And so we can use a tool called CompCaryo within RNA-Seq-Pop, which is a Python tool to predict karyotypes using karyotype tagging SNPs. And so this was developed by Rebecca Love and Nora Bozansky in this paper. And so we use CompCaryo within RNA-Seq-Pop to determine the frequency of common chromosomal inversions. And so this plot just shows a diagram of the sort of 5 chromosomal arms in Anopheles gambiae. And what we can see is that although the frequency of 2RB doesn't really differ too much between the two Busia strains, around 6% and 5%, so a little bit higher in Kisumu, the frequency of 2LA differs a lot between the two selected strains. So whereas it was around 35% in the Busia parental strain, this increased to around 86% in the Busia selected strain. And so that's interesting because it suggests that there may be an association with deltamethrin resistance. And actually, there has already been an association with deltamethrin resistance been found in Uganda in the past as well. So again, this is another nice result, which is kind of confirming what we've previously found before. And I mentioned FST was very high around the inversion. If we go back, this is also sort of capturing that effect where the frequency of inversion has changed a lot between these two strains. It's also worth noting that the CYP4J5 L43F marker actually sits within the inversion. And so it could be that that marker is not necessarily important for resistance, but it could be the reason why that marker has changed is simply because the inversion has changed frequency. And so another part of the pipeline is looking into ancestry. So as we saw in workshop 4, Anopheles gambiae sensilato is a species complex of 7, probably more, sibling species. And obviously the ones that we typically focus on, because they're the most medically important, are Anopheles gambiae, Anopheles colucii, and Anopheles rabiensis. And so the pipeline can use ancestry informative markers, which we met in workshop 4, to calculate the proportion of ancestry from either gambiae versus colucii, or Anopheles gambiae and colucii versus Anopheles arabiensis. And so in this plot, I've now colored each ancestry informative marker by whether it's an Anopheles gambiae allele or whether it's an Anopheles colucii allele. And so you can see that the ancestry informative markers themselves are quite sporadic across the genome. So they're concentrated in specific areas, such as in the beginning of 2L and at the end of the X chromosome. But the interesting thing that we found here is that although both Busia strains do indeed look like Anopheles gambiae, so 96% of the ancestry informative markers were indeed Anopheles gambiae alleles, when we looked at Kisumu, which should also be Anopheles gambiae, we actually found quite a different pattern, where although the X chromosome did indeed look like Anopheles gambiae, the autosomes had a lot of Anopheles colucii ancestry. And so it looks as though Kisumu is actually now a bit of a hybrid strain, actually, between the two species. And we've looked into this subsequently, and we've actually seen similar things in whole genome sequencing data, and also in other RNA sequencing data sets with different colonies of Kisumu. And so this is quite interesting, and it suggests that at some point in the past, I mean, it's a long history, Kisumu, around 50 years almost in the lab, but at some point it seems that it may have been contaminated with a different strain. And so this could also potentially explain why we're seeing resistance markers, which shouldn't, in theory, be in Kisumu, present in Kisumu. So to conclude, so a very brief overview of sort of the features of RNA-Seq-Pop. But RNA-Seq-Pop is a reproducible pipeline for RNA-seq analysis, which bridges the gap between our transcriptomic studies that we're doing and our whole genome sequencing studies. And so, despite the caveats that I've mentioned, I hope I've shown you that calling SNPs in RNA sequencing data is useful, and we can learn a lot more about our samples and our strains by doing so. And we hope that others may want to use RNA-Seq-Pop on their own data as well. So just to acknowledge my supervisors, Martin and Dave Wheatman, Ambrose Aruni, who helped with the establishment of the colony and the testing, and also Naomi and Eric Lucas for testing the pipeline. And thanks to all the people who developed software that has contributed to RNA-Seq-Pop. While we're on the subject of transcriptomic studies, I just wanted to highlight a new tool that I've developed called AnoExpress. And so this is basically a meta-analysis of all the published studies of RNA sequencing, all the published RNA sequencing studies that are investigating insecticide resistance in Anopheles gambiae and Anopheles funestus. And so this tool basically gives you an easy way to visualize gene expression for genes that you might be interested in. And so I just wanted to flag that here. But yeah, thank you very much. If you guys, of course, any questions now, very happy to answer, but also in the future, please just message me on the Slack channel, or indeed you can email me as well. Thank you.
Thanks, Sanjay. That was absolutely amazing. That's so much amazing work, all packaged in a very nice way for other people to use. Thank you so much for walking us through that. It's all amazing work.
Thanks, Anastasia. Very kind.
Does anyone has any questions or comments for Sanjay? I can jump in. So I have a very basic question. So as you were saying, like with RNA-seq data, we obviously only look at the coverage, and we can only look at the coverage in the express regions. So I'm wondering how that affects stuff like when we try to estimate allele frequencies, for example, and how that correlates with what we see when we use DNA. And I'm wondering if you did any kind of like correlation studies to see how that looks in your data.
Yeah, so I think so one of the things that like I wish we'd have done when we were developing RNA-Seq-Pop is to do like matching RNA sequencing and DNA sequencing on the same samples themselves, and then it would have enabled like a really like thorough analysis of that really. But unfortunately, basically, the original plan when we were doing RNA-Seq-Pop was not to develop a pipeline at all. It was just to do an RNA sequencing study. So we kind of started doing the RNA sequencing first, and it's only then after we had that original data, I started developing the pipeline and started SNP calling and everything. So we haven't thoroughly investigated it. One of the really positive things that we see is like what we kind of the allele frequencies that we tend to see do match up very well, roughly speaking, with what we see in whole genome sequencing studies. So where we have cohorts from the same country or the same regions, we do see, we see the markers in the places where you would expect to see them, which obviously does give confidence, and also to generally roughly the frequencies that you would expect to see. So yeah, we haven't done a completely thorough analysis of that, but mostly because what we see makes a lot of sense, that kind of adds a lot of confidence, really. Yeah, I do think that would still be a good thing to do, a study to do in the near future, though, definitely.
Thanks. It's very, very clear. Any other questions from anyone else? Please feel free to write in the chat, or you can also unmute yourselves. There's a question from Sonia. Please go ahead, Sonia.
Thank you, Anastasia, and great presentation, Sanjay. Just wondering, my question is a little bit naive though. If what would it take to reproduce this pipeline if, say, somebody wanted to run the same analysis on a different organism?
Yeah, thanks, Sonia. Yeah, so the idea is that it would be, like relatively straightforward, essentially. As I mentioned, like the pipeline can be applied to any organism. So yeah, so it's just a matter of configuring the workflow. The things that you need are obviously the RNA sequencing data itself. You have a metadata file just to tell the pipeline what each sample is, basically. And then kind of your reference genome files. So one thing at the minute you do need is like a reference, like a relatively not high quality reference genome, but ideally one which has sort of chromosome level like data, like Anopheles gambiae and most of the organisms that we work with. But yeah, shouldn't be too much. You just need sort of the reference genomes, the reference transcriptomes, the kind of standard things you can get from vector-based, basically. And then your input data and a metadata file. And yeah, there is on the documentation site, there is like a video walkthrough of how to set up RNA-Seq-Pop, and also like documentation in general. And also I'm happy to answer specific questions whenever people are sort of struggling with it. Thank you.
Sonia, any other questions?
Yeah, Trevor has one in the chat. which says, for the proportion of the SNP calls that fall within the intergenic regions, are they useful in downstream analysis? Yeah, so they are used in things like, so yeah, certainly. So if there's a few reasons when it'll be used. So for example, for the genome-wide selection scans for the FST, those SNPs will be used that are in intergenic regions. Also, if any of those SNPs happen to be ancestry informative markers, we would use them in the analysis for ancestry informative markers. So yeah, they would still be used, the intergenic SNPs, basically in most situations, I think. There's a few things which I didn't talk about in the pipeline that it does, which are things like for each gene, I kind of forgot actually, each gene it will calculate, as well as doing FST across the genome, it will calculate FST between each gene as well. So for between each condition within a gene. So you can sort of rank genes by how high FST is, which is sort of another nice additional bit of information. But generally, yeah, the intergenic SNPs will also be used for most of the downstream analyses.
Thank you, Sanjay. That clears it up. Thank you.
Thanks, Trevor.
Any last questions or comments for Sanjay? Last one from me. So it's on Anno Express. So are you planning on adding more information, more RNA sequencing studies into it, or is it like have you like caught the meta-analysis, or are you gonna kind of like keep maintaining it?
Yeah, no, so the idea was to every so often to add new data sets into it. So at the minute I think there's like 35 different at least different differential expression comparisons, I think from around like 25 studies or something. And yeah, the idea, we haven't added any more yet. I think we're going to try and get initial manuscripts out, and then after that, probably, you know, once every year, add in a few studies. And so yeah, on the GitHub, if anyone does have RNA sequencing data that they'd like to contribute to it, we go just from the raw read counts. We don't go from like the FASTQ files, just for convenience, really. But yeah, you can just email me or raise an issue on the GitHub, and I'll be happy to include that data.
Awesome. Thank you so much.
Thank you very much, Anastasia.
Yeah, so if we don't have any other questions or comments, I'll just say thanks again to everyone for joining us today, and especially to Sanjay for an amazing talk. And yeah, I think that's everything we've got for today. So please take care, and I will see you on our next workshop on Wednesday. Thanks all.
Awesome. Thanks very much, everyone. Thanks, Anastasia. See you later.